Yeah. In your background, you took a small molecule, as I understand it, from concept to clinic in roughly 3 years at Silicon Therapeutics, targeting Sting, which is known to be an extremely difficult target. Can you talk to us a little bit about the engineering or computational scaffolding that made that timeline possible?
Yeah, so that was a great program because it it did really combine many aspects of the computational and wet lab capabilities and all the accumulated learnings over the years. So Sting, we were looking for a Sting agonist, which is basically something that activates a pathway. So a lot of drugs that people think about are antagonists that block a pathway, which it ends up that there's a lot of ways that you can mess up a pathway. It's a lot harder to actually activate the pathway. And so that was one of the key challenges, and it was a motion-based thing. Sting is a flexible protein that undergoes a conformational change in this activation process. And so we had to not just bind, but bind and lock it into a conformation that was productive for signaling. And the third bit that was particularly hard there is that the endogenous molecules that bind to Sting are cyclic dinucleotides, and these are big, ugly molecules that don't really have drug-like properties. And people historically that have worked on Sting have started with these cyclic dinucleotides, and they could find molecules that worked fine in a cell, but they just didn't work as drugs. They didn't have drug-like properties. They got cleared out of the body before they could actually do anything to the target. And so we took a very novel approach. We found a small molecule that was much smaller than these cyclic dinucleotides that had drug-like properties, and we got it to dimerize on itself, bind to itself through a quantum mechanical pi-pi stacking interaction, which I don't think has been done before. And so in order to do that, we had to run a lot of quantum mechanical simulations, designing molecules that enhanced this intramolecular interaction. We had to run a lot of molecular dynamic simulations, understanding how the protein moves and in the context of these molecules binding. A lot of structural biology and biophysics, a lot of integration with the lab, the iterative design, make, test cycle, and building an organization where all the people were bought into this idea. Computation can be super powerful when used appropriately, but you can't make drugs on computers. You can come up with ideas, you can triage vast chemical space. Hypothesis generation is super powerful, but designing that design, make, test cycle still requires making and testing in the lab. And so building that company from scratch with the right people that were experts in drug discovery who were fully bought into the computational platform approach was super important as well. And so there's a lot of technical and scientific aspects in the platform that made it possible. There's also the human kind of psychology and behavioral team dynamics that came together really nicely. And we were able to crack this nut that I think is still unprecedented for designing a small molecule human Sting agonist.